kind of the weak links in those chains, which would be not the eccentric contraction, probably the concentric contraction. And then we're testing out the strength of that muscle at probably when it's fully stretched out, or maybe when it's already fully shortened, one of the two. And we don't necessarily need to differentiate between which of those two would be weaker, but we know that the midpoint of the range of motion is the strongest, and we know that the eccentric contractions are the strongest. And part of this is just natural for us, right? Because if you think about if you're going to do something like a jump, right? So you want to just jump up as high as you can. You're not going to just bend your knees a tiny bit and then just try to jump up. You're also not going to put your butt all the way on the ground and then try to jump up. You're going to start in a position that's somewhere right about in the middle, right? And a lot of that is having to do with the length tension relationship, right? And so that's just kind of an application of those ideas. So any questions on those things before we move on? Cool. And then let me actually introduce somebody to you who's on the Zoom with us today. Some of you already know her. Sophie Murth is there. Let me pin her for a second. There she is. I think she probably hopped up on the top of your list there. But yeah, so Sophie is an alum from Point Loma. She graduated last year, and she is now a grad student with us in the MS Kin program. And she's working with me in a couple different ways, doing a thesis with me, but then also doing an internship, kind of like a teaching internship with me. So she's going to be sitting in on my classes, and then also we'll be kind of, she'll actually teach a class or two in this class this semester, because it's her goal to get her PhD and become a professor. So it's a great opportunity for her to get a little training in that while she's here as a grad student. So just want to introduce you to her. She'll also be grading some of the stuff that we do this semester. So you can get mad at her and not me when she she grades things. Yeah. Anything you want to say? So.
Hey, I'm excited to be here.
Cool.
Yeah. See you all in person next week, hopefully.
Perfect. Thanks. Okay. Cool. All right. Let me swap that back so you can see. There we go. Oops. Okay. All right. So yeah. All right. Well, let's go ahead and move on. So we're gonna dive into kind of more how muscle cells actually contract now. And so before we do that, we need to at least kind of mention the fact that this is done by the nervous system, right? The nervous system has to be involved in this. We're going to talk about the nervous system more in the next section, and just in the next at the end of today and then Friday as well. But it's this whole idea that we have neurons coming down, and those are going to activate the muscle cell. Okay. One interesting thing when we think about the setup of the nervous system, we think about like a nerve, right? With these nerves we can touch, like our ulnar nerve and our sciatic nerve. Those are all axons, right? All that we find in those nerves is a bunch of axons. Okay. When we think about the cell bodies up here, those are all in the central nervous system. Okay. So those are pretty much all in the brain or the spinal cord, with the exception of a couple ganglions in different places, like right outside the spinal cord, we have these little ganglions, sensory ganglions. But for the most part, so all these motor neurons that we talk about, the cell body of those is actually all the way back in the spinal cord. And then what we get actually going down to the muscle and all the way between the muscle and the spinal cord is axon, right? So these extremely long cells, but all those cell bodies are in the central nervous system. Okay. And we talked a little bit about it last time, but this language of communication between the muscle and the brain, what do we call that signal that goes down an axon there? It's called an action potential, right? So we get action potential. And I know my handwriting is not great, and it's always a little trickier for me on the iPad. So if at any point you don't know what it says, just please unmute yourself and ask me. I don't take any offense to that at all. Yes, my wife says our 5-year-old writes better than me, so it's totally okay if you say, please tell me what the heck that word says. Okay. Yeah. So then the question of what do these action potentials travel on? They travel on these axons that we're talking about that are going to make their way down to the target tissue. And in this case, what we're talking about, skeletal muscle. Okay. So once that action potential gets down to the skeletal muscle, then it gets to this place called the neuromuscular junction, or we'll just abbreviate it as NMJ, right? and the primary neurotransmitter in the neuromuscular junction, put that in the chat for me. Put the three-letter abbreviation of what that is. What's the neurotransmitter in the neuromuscular junction? It's always the same between skeletal muscle. There we go. You guys remember that? ACH, right? Acetylcholine. Okay, good. So it's going to be acetylcholine. Okay, so that neuron is going to release acetylcholine, and that's going to cause some things to happen. We'll talk about those things here in a sec. Okay, and the thing that happens immediately when the neurotransmitter binds to the muscle cell, that causes that muscle cell membrane to depolarize. Okay, so if we got resting membrane potential of about -70, depolarization means that it goes up in a positive direction. Okay, so it goes from -70 up to about +55 for an action potential, yeah, and so that's what happens. So that's really the whole effect of the nervous system, is to depolarize the membrane of that muscle cell, and then we can kind of go from there as far as what's happening from there. Okay, questions so far on any of that? Okay, so yeah, let's yeah, I'm not going to send you to breakout rooms for this one, but just go ahead and think about this one for a sec. And in about a minute, I'll tell you when to hit enter on that chat, and you can put in your answer there. Okay, so think about this one for a sec. Transverse tubules, what is their role in muscle contraction? Also known as T tubules. So you can have the letter in the chat, just be ready to hit enter, just not quite yet. All right, go ahead and hit enter. There we go. Okay, so we're kind of mostly C's, a couple of B's. Yeah, but mostly everybody's saying C. So C would be to allow the action potential to spread from the cell membrane to the sarcoplasmic reticulum. Perfect. Okay, and so like we just said right here, the neurotransmitter causes depolarization of the membrane. Okay, and then if we get enough depolarization in this region, this is what's called a local potential or a graded potential there in that region. If we get enough depolarization, then it's going to spread out here, and out here we can actually get the action potential. Okay, because we actually don't have those sodium channels, the special sodium channels and special potassium channels that are voltage gated. We don't have those in here. We have some different ones. So we get graded potentials until we get out here, and then we have those sodium channels that we'll talk about a little bit on the outside of the neuromuscular junction, and that's where we can actually have an action potential. Okay, so we get that action potential, it spreads across the muscle cell, but the problem is muscle cells are massive. Okay, they're absolutely huge. When you look at a muscle cell next to a red blood cell, muscle cells are about 100 times the thickness of a red blood cell. Okay, so what that means is we've got all this sarcoplasmic reticulum on the inside of that muscle cell, and if we just had the action potentials going along the membrane, none of the sarcoplasmic reticulum on the inside would be affected by that. Right? It wouldn't have any effect on it. We would only be able to activate the sarcoplasmic reticulum on the outside of the muscle cell, and that wouldn't be very efficient. So we need a special structure in the skeletal muscle to get that action potential down deep to where it can activate all of the sarcoplasmic reticulum, and we can get a lot of calcium release. Okay, and so that's kind of what this is showing here, right? So if the action potential just stayed up here on the membrane, we would only really activate this very perimeter of the muscle cell, right? So we need these special structures to dive down deep into the muscle cell to activate all of this sarcoplasmic reticulum here. Okay, and this is kind of looking at the T tubule right there. Shows where that action potential will go down. There's these special little receptors that connect to the T tubule membrane from the sarcoplasmic reticulum. When that voltage change hits them, it leads to them opening their calcium channels and dumping calcium into the muscle cell, into that sarcomere, where then it can do its job. Okay, and what its job is it's going to go bind to troponin. So where does it come from first? It comes from the sarcoplasmic reticulum. Okay, and how does it allow action potential to occur? Well, the calcium binds to troponin. Okay, so we'll say binds to troponin. Okay, troponin changes shape. Okay, and this kind of takes us back to anatomy and physiology, probably cell biology as well, when we talked about how proteins are made, right? So we have the primary structure of a protein, secondary, tertiary, quaternary. Nod your head if that sounds somewhat familiar. Yeah, for sure, right? And so that primary structure is all covalent bonds, right? Those peptide bonds are really strong. We're not really going to change those very easily. We need enzymes to break those bonds apart. But once we get up above the primary structure, we get to the secondary structure, the tertiary structure, the quaternary structure. Most of those bonds, most of them are hydrogen bonds, right? Which means maybe a polar molecule interacting with water, right? If there's an amino acid that has a polar side to it, a polar R group, then it's going to stick towards that water, right? If it's a non-polar R group, then it's going to move away from that water, right? If we have one amino acid that has a positive charge next to another one that has a negative charge, those are going to bend towards each other, right? And so this is exactly what happens with calcium. On the troponin molecule, we've got all these different amino acids. There's a binding site for calcium. So that means there was probably a negatively charged amino acid there, and it can bind to this +2 calcium charge. And now that amino acid is no longer negatively charged. So that means it's going to interact with the ones next to it differently, right? It's going to interact with the water differently. It's going to interact with the pH of the solution around it differently. And that completely changes its shape, right? And that's the cool thing about biochemistry, is that in this class, we get to dive into it a little bit just with things like calcium binding to troponin, the myosin head shape change as well. But so all those amino acids have to be right in the perfect spot to where there's a nice little binding site for a positively charged molecule or a positively charged ion like calcium. And when it binds to it, it totally changes the way that that amino acid interacts with its surrounding environment. So it totally changes the shape of troponin, right? And it just so happens that troponin is connected to this other one called tropomyosin. Okay, so here's troponin C, which is the one that calcium binds to. That's the TNC one there, okay? And it just happens to be connected to tropomyosin, okay? So when troponin changes shape, it moves tropomyosin out of the way, okay? So first step, calcium binds to troponin, and then that changes its shape. Second step, troponin, by its shape changing, moves tropomyosin. Right, so it just pushes tropomyosin out of the way, and now we have this open binding site on actin that is ready to have myosin bind to it. And I'm not sure if you could hear that or not, but it's not me snoring during class, it's my dog. So she's back there on the couch. But Casey thought I was like falling asleep or something. I'm not. But yeah. Okay, so that's what happens. So we move tropomyosin, and then what this results in is an open binding site on actin for myosin. Right, so that binding site on the actin is now open, so the myosin head can bind to it. Okay, questions on that? So all of this really is called excitation, contraction, coupling. I should have given myself a little more room for that. But yeah, it's all, we can just call it ECC, excitation, contraction, coupling. That's what this process is called, just preparing for contraction. Let me read it again. Excitation, contraction, coupling. Okay, questions on any of that? Anything you need clarification on? Okay, all right, so now that's pretty much our preparation for contraction. What I'm going to do next is show you this little video that goes through this process a little bit, and then also goes through the cross-bridge cycling process. And what I'd like to do after that is send you to your breakout groups and have you work through this slide together. Okay, so this just kind of goes through the steps in this, like cyclical steps in the process of cross-bridge cycling, which is what happens for us to shorten muscle. Okay, And so after we watch this video, I'm going to send you to your breakout rooms, and pretty much I want you to fill in what's happening in each of these steps. Okay, and we can go ahead and number these. That way, that'll help you answer those questions on the right. Okay, so you can answer each of these questions as well in your group when I send you out. Okay, and then yeah, all right. So let's take a look at this video, and let's see. Just give me a thumbs up if the sound
Muscle
is coming through for you.
contraction is at the
Yeah,
basis
thank
of
you.
all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive.
The last part. Okay. All right. So this this is just from a actual scientific article that measured the distance that the myosin head moves when phosphate binds to it, and then when phosphate is removed from it. And so they actually measured it to be 100 angstroms, which I have no idea exactly what that is, but I guess in molecular terms, that's a pretty large amount of movement. When I read this article, they were discussing how that's a pretty large conformational change in a protein. Okay, and it's just kind of amazing to think that this is how all organisms across the spectrum of life move, right? Almost all organisms use some form of this kind of contraction to move themselves, right? And so it's just all about the fact that all of the little amino acids that make up this protein, they're in such a perfect order that, again, there's a perfect binding site for phosphate. And so when phosphate binds to it, it completely changes the properties of that amino acid to where it completely changes the shape of that protein, cocks it, and then the phosphate comes off and it completely changes the shape back, so it moves us, right? But this has to happen for any organism to move throughout the whole spectrum of biology, which is, yeah, pretty cool kind of commonality between us. Okay, so that's that. So let's talk a little bit about different muscle fiber types, okay? And you guys are familiar with these. One thing to remember, I will just go to this slide, I'll come back to that one in a second, but the naming of the fibers is based on two things, okay? It's based on the, like I just mentioned, the type of myosin ATPase bound, okay? And that's just going to be the fast or the slow, okay? So there's just two types, okay? And then also the energy system primarily used, okay? And that's either going to be oxidative or glycolytic. And we'll get into those energy systems and that a lot more next week when we talk about metabolism. But so the three fiber types are going to have either fast or slow in their name, and they're going to have either oxidative or glycolytic in their name. Okay, so let's go back to that. Well, I'll leave this up for a second. I see some of you are still writing that down.
What does it say? ATPase and then
ATPase found, so type of myosin ATPase found, and then below that it says fast or slow. Thanks for asking, Naomi. Other questions on those? Okay, so then let's go back here. So then when we look at these three fiber types, we've got type 1 and then type 2, but underneath type 2, we've got two subtypes, type 2A or type 2X, okay? And so type 1s are called slow twitch, so they have the slower myosin ATPase, and they primarily use the oxidative system, okay? These are our endurance fibers, right? So these have a lot of endurance. They don't produce a lot of force, but they can produce a lot of force, or they can produce a little bit of force for a long time, right? So yes, not a ton of power, but they can do it for a long time, lots of endurance. Okay, type 2A fibers are the ones that are kind of in the middle. They have the faster myosin ATPase, but they tend to use more of the oxidative system. So they have more mitochondria inside of those muscle cells than the type 2X fibers. Okay, but they also have the faster myosin ATPase. So these are kind of our middle distance type fibers. So these are definitely good for sprinting type activities, but more sprinting kind of, greater than 30 seconds type thing, but really it's more than that, greater than 10 seconds. But when you think of an event that really exemplifies the use of these type 2X, or sorry, these type 2A fibers, would be like a middle distance event like the 400 or the 800, something like that, that lasts somewhere between 1 to 2 minutes. Okay, they're going to be used for lots of things, but when we really want to exemplify how they're used, probably be those middle distance athletes. Okay, and then when we talk about the type 2X, these are mostly used for kind of just power type movements. Right, so we're thinking about a vertical jump, right? We're thinking about one block thrown by an offensive lineman, right? We're thinking about a very short sprint, like a 100-meter dash, or like 50-meter dash, something like that, right? Thinking about just single muscle contractions when we're doing the bench press, right? Something like that. So just quick, powerful movements that occur. Those are really exemplify the use of the type 2X fibers. Okay, questions on that? All right. Oops. Okay, so the way that we identify muscle fiber types is based on this here, which would be a muscle biopsy. Okay, so muscle biopsy uses this big hollow needle, and it's typically in different states. There's different policies on who can do these, but in a lot of states, it's only a physician has to be present, partially because you have to use a scalpel and slice through the skin, slice through the connective tissue to get down to the muscle, and then put the needle in, and it just fills up with muscle tissue, right? When you have that hollow needle and you put a little suction on it, and then that pulls out some muscle tissue, put that into a microscope slide, put some different types of stains in it, and then you get something that looks something like that. This was actually a couple of images. This image and this image were sent to me by one of our alums, Steph Gagnon, who just finished her PhD at a university in the UK called Loughborough University, and now she's doing a postdoc at Stanford with a neuroscientist actually, but she was doing a lot of muscle biopsy research during her PhD, and this was some of her slides that she sent. So yeah, so this is another type of staining that we can use that would give us all three muscle fiber types here, and you can see the darkest ones are the type 1 fibers. Those are red. Okay, the ones in the middle are kind of pinkish, those type 2A fibers, and then the ones that are white are the type 2X fibers. So part of that is based on the amount of myoglobin inside of muscle cells. When they have a lot of myoglobin, that means they deal with a lot of oxygen, and that myoglobin gives it a reddish color. And so those are the more oxidative ones. So the dark meat in turkey is always the type 1 fibers, whereas the white meat is the type 2 fibers, type 2X. Okay. Questions on any of that? Okay, and this just kind of walks us through some basic characteristics of each muscle fiber type, right? And you'll notice some patterns here that typically type 1 and type 2B in this case. So it's called B when they deal with it in rats, and that's why it's B. But when it's in humans, it's type 2X. That's why the only reason there's a difference there. But what you'll see is that those two are always on the extremes, and the one in the middle, the type 2As, is kind of the mid-range one, right? So when it comes to diameter, force, rate of contraction, myosin ATPase activity, we see that type 1s are the slowest or the smallest, whereas these type 2Bs or type 2Xs are the fastest or the largest, most powerful, right? But then when it comes to mitochondria, fatigue, oxidative phosphorylation, it's just the opposite, right? And then we got glycolysis there, which would be the opposite again. And then this one is pretty much mostly in the middle for most of them. So just kind of a general look at those different characteristics there. Okay, any questions on any of that? Okay, so we're going to start with just a little bit of an intro of the next section. And so I'm going to pull up the notes on the nervous system here. Okay, so we'll just start out with a little bit of this for the last 10 minutes, and then we'll continue this and finish it up on Friday. Okay, so we kind of already talked about this, but this whole idea and this anatomy of the nervous system really dictates that all the cell bodies of neurons are found in the central nervous system, either the spinal cord or the brain. And then when it comes to the peripheral nervous system, it's pretty much all axons, right? So axons out here and somas or cell bodies in the central nervous system. Okay, and so one of the things that we really need to wrap our heads around is this idea of action potentials and how they work. And so we're going to spend some time doing this. This is review from Anatomy and Physiology, but I know some of you took it a long time ago. This is one of the harder concepts at the end of Bio 1030 for sure. So it'll be a good review. But yeah, so yeah, when we look at this image here, this is the inside of the cell here. Okay, this is the outside. Okay, so in the chat, what I'd like you to send in is which ion do we find a lot of outside of cells? Well, let's do inside, okay? Which ion do we find a lot of inside of cells that we don't find a lot of outside of cells? Send that in the chat. Okay, John's right in there. Okay, yeah. Okay, so most of us are saying potassium. For sure, okay? So potassium is known as the primary ion of the intracellular space in our bodies. Okay, we got lots and lots of potassium inside of our cells, but not much outside, right? We got a lot inside, not much outside. And then which one is the flip of that? Go ahead and put that one in the chat. Which one do we have a lot of on the outside, but not much on the inside? Yeah, so most of you are saying sodium for sure, right? So we got lots of sodium outside, not much inside. Okay, and those two things are crucial to understand, right? That's what during the resting state in skeletal muscle or in a neuron. This is what it looks like. We got lots of sodium on the outside, lots of potassium on the inside, and it's because of those concentration differences that we can have something like an action potential. Without those concentration differences, we wouldn't be able to have action potentials. Okay. So there's a few things that help us to make sure that the inside of our cells stay negative. Okay, so the inside of the cell is typically around negative 70 millivolts, right? That's resting membrane potential. We can see it up there in that graph. Okay, and so the question is, why is it negative on the inside, right? What things make it negative on the inside? And there's three primary reasons for that. Okay, so which side is positive? We'll say outside. Which side is negative? Inside. Okay, so reason number one for why it's negative is this guy right here. This is called the sodium potassium pump. Okay, what it does, as you can see here, is it pumps 3 sodiums out and 2 potassiums in. Okay, so both of those are going against their concentration gradient, right? Because we've already got a lot of sodium outside, we've already got a lot of potassium inside. So that is called active transport, right? When we're pushing something against its concentration gradient, it's active transport. And when we're using ATP to do that, it's called primary active transport. Okay, we'll talk about some examples of secondary active transport later, but when we're using ATP to push something against its concentration gradient, it's primary active transport. Okay, but you can see that they both have a +1 charge. So if we're constantly, and we got tons of these on our cells, okay, tons of these little sodium potassium pumps. So if we're constantly pumping 3 pluses out and only 2 pluses back in, what's the net charge change with that? -1, right? Yeah. So this is constantly causing, every time it pumps, it's causing a -1 charge change to the inside of our cell, right? Because we're losing more positive than we are gaining positive. Okay, so that's reason number one for resting membrane potential being negative is a sodium potassium pump. Okay, reason number two is because we have more potassium leakage than we do sodium. Okay, so when it comes to leakage, there's actually special little proteins on our cell membranes that specifically allow certain ions to leak. Okay, and so we can see those right here. That's That is a potassium leakage channel. This would be a sodium leakage channel here, okay? And so since there's a lot of potassium on the inside, not much on the outside, if it's just going to passively leak, it's going to go out, right? It's not going to come in, it's going to go out. But with sodium, it's the opposite. When sodium passively leaks, it leaks back in, okay? So if we have more, if we're losing more of a positive charge because more potassium is leaking out than sodium's leaking in, that's another negative on the cell, right? That's another thing that causes our cell to be negatively charged. Okay, so that's reason number 2. Reason number 3, whoops, is we have negatively charged proteins inside of cells. Okay, our cells are full of a bunch of proteins. Many of those have a negative charge to them, and those aren't moving back and forth. They're too big to move back and forth. And so those are the 3 reasons why we typically have this negative resting membrane potential. Okay, questions on any of that? Okay, so another thing to be aware of is when we're going to talk about sodium rushing across the membrane soon. We're going to talk about potassium rushing across the membrane. One thing to be aware of is that we need to think of both the chemical gradient and the electrical gradient when we talk about how fast those ions are going to rush across the membrane. Okay, so when we all of a sudden open up gates for sodium to rush in, first of all, we know it's going to rush in because there's more on the outside than there is on the inside, and that's its concentration gradient. But which way is sodium being pushed by the electrical gradient? Go ahead and put that in the chat. Just say in or out. Which way is sodium being pushed by the electrical gradient, in or out of the cell? Absolutely, right? Everybody's saying in because the inside's negative, sodium's a positive charge, opposites attract, right? So sodium not only has a strong chemical gradient to push it in, but it also has a strong electrical gradient. And that's why so much of it rushes in during an action potential, okay? We need potassium to flow out too, right? But when it does that, it's going against its electrical gradient, right? Luckily, it has a strong enough chemical gradient to allow it to push out. But it is going against its electrical gradient when it does that, because that negative charge of the inside of the cell is kind of pulling it back, right? So that makes it so not as much potassium moves as sodium does, and at least doesn't do it as rapidly. Okay, yeah, and so I think that's kind of a good starting place. Or stopping place. We'll come back to this next class where we talk about the actual steps in an action potential and what happens with these little gates and that we find in the sodium and the potassium channels. All right, guys, great to be with you again, and we'll see you on Friday for the last class of the week. Hopefully, the last class online.
Thanks, Dr. Sawyer.
Yeah, see you guys.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you.